My name is Anjali Vinocha, and my co-author on this presentation is Michael Lee. We are from MD Anderson Cancer Center. We will be presenting our poster on the role of women in cytogenetics. Genetics is the scientific study of genes, and cytogenetics is a further branch of genetics which is focused on the study of chromosomes and their inheritance. Figure 1 shows the normal karyotype. A karyotype is a phenotypic presentation of individual's complete set of chromosomes. Like in many other fields, women are underrepresented in the field of cytogenetics. Despite all these obstacles, women scientists have made major contributions in numerous scientific fields. As depicted in Figure 2, according to a study by UNESCO, less than 30% of the world researchers are women. This section of the figure highlights women in leadership roles, with only 16% serving as dean, 18% as department chairs, and 25% as full-time professors. The objective of our study is to identify and offer a concise overview of the significant contributions and achievements made by women in the field of cytogenetics. Regarding the methodology, we have gathered and compiled information from various validated databases and published literature sources on accomplished women in the field of cytogenetics. Now over to Michael.
Thank you, Anjali. From the gathered data, we see countless women scientists making contributions to cytogenetics. We have selected a few women scientists and created a timeline that can be seen in Figure 3. This timeline can be broken down into 3 different eras. Let me highlight a few from each era. The first era helped establish the basic understandings of chromosomes. It began with Nettie Stevens' discovery of sex chromosome in 1905, after Lillian Morgan discovered attached X chromosomes and ring X chromosomes in 1921 and 1933, respectively. Last, we have Barbara McClintock discover mobile genes within chromosomes in 1948. The second era came with those that link diseases to the chromosomal abnormalities. In 1959, Martha Gautier found that Down syndrome individuals have 3 chromosome 21s. Within that same year, Patricia Jacobs identified the presence of an additional X chromosome in males with Klinefelter syndrome. Last, in 1961, Joy Delhanty discovered the first human with 3 sets of chromosomes, later known as triploidy. In the most recent era to today, we came into a period of advancements made to view chromosomes at higher resolutions. In 1968, Lortzek established the first chromosomal staining technique called cube bending. She worked on this along with Casperson. However, her work was overshadowed by him, causing her contributions to be less known. A year after, Mary Lou Purdue came up with fluorescent in situ hybridization, which helps identify and visualize specific DNA sequences on chromosomes. In 1971, a woman scientist named Marina Seabright created another staining technique called G-bending. This technique, till today, remains the most popular bending protocol. The women scientists I just mentioned are only a few of the many who have shaped the advancement of cytogenetics and science overall. Some women scientists' work may have gone unnoticed and unrecognized, so we want to pay tribute to all women scientists by showcasing some of their portraits in Figure 4. We hope by highlighting some women scientists' work and achievements, we inspire the next generation of women to pursue careers in cytogenetics and other STEM-related fields. In closing, we want to say thank you for the support given to us by our Dean, Dr. Crumweed, and other professors, Dr. Baker, Dr. Hill, and Dr. Manchu. Thank you for listening in.